So I would like to thank the organizers for this opportunity to present our work. CCR6 is a well-established marker for Th17 cells. CCR6 mediates migration into specific sites, including the brain and the pair patches of the small intestine. Studies by our groups and others demonstrate that CCR6 is a marker for CD4 T cells highly permissive to HIV infection in vitro and in HIV subjects. CCR6 is also a marker for HIV-specific CD4 T cells. Studies by the group of Sharon Lewin demonstrate that signaling via CCR6 induces HIV latency in resting CD4 T cells. And of particular importance, the group of Ashley Haas demonstrate that inhibition of the CCR6 CCL20 regulated cells trafficking via glycerol monourate prevents SIV dissemination upon mucosal challenge. So all of this evidence supports the major role of CCR6 positive T cells in HIV pathogenesis. So as you all know, the gut is a major site for HIV replication. Expression of gut homing molecules such as integrin alpha-4 beta-7 or CCR9 on immune cells is induced by retinoic acid. Retinoic acid is a metabolite of vitamin A produced by intestinal dendritic cells. In fact, retinoic acid binds to its receptor and regulates this way the expression of numerous genes after interaction with the retinoic acid responsive element site. Of note, such elements are present in HIV LTR, and then retinoic acid may regulate directly HIV transcription. Studies by our lab demonstrate that treatment with all-trans retinoic acid, or ATRA, induce the expression of integrin alpha-4 beta-7 in both subsets, CCR6 positive and negative T cells, but an increase in HIV DNA integration and replication was observed only in CCR6 positive T cells after ATRA treatments. This effect is at least in part explained by the upregulation of the HIV co-receptor CCR5, but exposure with a single-run VSVG pseudotype HIV indicates that ATRA preferentially induce HIV DNA integration in CCR6 positive T cells. So this result are providing the fact that ATRA facilitates HIV replication in CCR6 positive T cells by acting at both entry and post-entry level. So the objective of this study is to identify and functionally characterize new molecular determinants of HIV permissiveness versus restriction in CCR6 positive T cells versus negative with gut homing potential. So to this aim, we use the Illumina technology for genome-wide analysis of gene expression in CCR6 positive and negative T cells, treated or not with ATRA. We identify approximately 2,000 genes modulated in both subsets with a p-value lower than 0.05. With a fold change cutoff of 1.3, we identify gene regulated by ATRA only in CCR6 positive T cells or only in CCR6 negative, as well as the gene modulated in both subsets. So we can say that in 180 gene regulated in CCR6 positive only after ATRA treatment include some dependency factors. Intelligent search for transcripts involved in cell homing, we confirm that ATRA induces the expression of CCR9 and integrin alpha-4 beta-7 in both subsets. We also identify CCR6, a minor HIV co-receptor, as being upregulated by ATRA in CCR6 positive only at the mRNA level and protein level. Further intelligent search identify for transcripts involved in cell activation and HIV permissiveness, demonstrate that ATRA induce the expression of RIGI and CD38 in both subsets, but at superior level in CCR6 positive T cells. The ligand of CCR5, CCL3, and CCL3L1 were downregulated by ATRA in CCR6 positive T cells, suggesting that ATRA facilitates HIV entry via CCR5 in these cells. KLF2 and CD96 and LCK were specifically upregulated by ATRA in CCR6 positive T cells, while NFEL3, ABCA1, and PPAR gamma were downregulated. Gene set variation analysis identify pathway specifically modulated by ATRA in CCR6 positive T cells, including AKT and PI3 kinase involved in the Th17 pathway, and mTORC1 pathway involved in autophagy process. Despite the fact that different reports that ATRA induce the Treg differentiation, we observe that ATRA does not interfere with Th17 features in CCR6 positive T cells, including the ability to produce IL-17A. Because RORC is a master regulator gene for Th17 cells, we used AMAXA nucleofection technology for RNA interference against RORC. The decrease of RORC mRNA was associated with a decrease of IL-17A production, while no changes were observed for cell proliferation. So the silencing of RORC was associated with a decrease of HIV DNA integration in CCR6 positive T cells. We validate that ATRA by Western blotting that ATRA induce the expression and the phosphorylation of mTOR, suggesting that this pathway is altered. Though experiments are ongoing to further unveil mechanisms by which ATRA interfere with the autophagy process with the HIV relevance. So in conclusion, we have two subsets. ATRA modulate gene expression in two subsets, but the induce transcriptional program is distinct. In CCR6 negative T cells, we do not observe diminution of CCR5 expression, but an increase of HIV restriction factor, ABCA1, and NFEL3 involved in Th17 negative regulator of Th17 polarization. In CCR6 positive T cells, there is an upregulation of HIV positive HIV co-receptor CCR5 and CXR6, and also KLF2, a transcription factor involved in CCR5 expression. We observe a decrease of HIV negative regulator, PPAR gamma, and CCR5 ligand. And finally, the increase of mTOR phosphorylation suggests a decrease of HIV degradation via autophagy in ATRA-treated CCR6 positive T cells. So all of these results indicate that cut-homing CCR6 positive T cells are major HIV targets and may play a critical role in viral dissemination for the portal site of entry. So I would like to thank my mentor, Petronella Ancuta, the member of my lab, also the collaborators and the HIV positive and negative patient for their gift for leukapheresis, and you for your attention. Thank you.
Any questions? Oh, yep.
Are there other markers on the the CCR6 negative cells that account for some of the other differences? Have you looked at any other panel of markers on those cells?
I'm sorry, can you repeat it?
On the CCR6 negative cells, have you looked at any other markers that might account for some of their differential phenotype and function and their ability to respond to the ATRA?
In CCR6 negative.
I'm sorry, in the CCR6 negative cells, have you looked at a panel of other markers which might account for their inability to respond?
We do not observe any difference in the activation of the cells. We do not observe any difference in the cluster formation by microscopy. So it doesn't seem that ATRA can decrease the activation of the cells. If I respond well to your question, I don't.
Very interesting results. I was just wondering if in your hypothesis you consider that all the effects you observe, like the phosphorylation of mTOR, the transcription, etc., are they linked to the retinoic acid receptor, or some of them can be totally independent and targeted from all-trans-retinoic acid to another receptor or to other adaptators in the cell?
I think yes. If, we have we have an effect direct of ATRA. There is a known molecule regulated directly by ATRA, but by retinoic acid. But yes, there is also an indirect effect that we do not yet identify. But for sure, there is other. There is an indirect impact of ATRA on the modulated gene.
Is there a way to inhibit the interaction between all-trans-retinoic acid and the receptor?
I don't know.
Okay, thank you.
Very nice talk. Have you had a chance to look at CCR6 positive cells isolated from either the female reproductive tract or gut mucosa to see how the expression levels, patterns, and response to ATRA is in those cells from